Welcome to ANZ 2022. My name is Saikat, and I'm finally a PhD student in AIBN at UQ. Today, I would like to share with you some insights from my research project, which focuses on developing Immuno-PET agents for targeting glioblastoma. Among the different subsets of brain cancer, glioblastoma is recognized as the most aggressive and is associated with a very high mortality rate. It originates in the brain with uncertain risk factors. Due to its infiltrative nature, it is highly recurrent and resistant to treatment. Patient survival time is about 15 months, and unfortunately, this statistic has not improved in the last 35 years. Current standard of care for glioblastoma involves maximum surgical resection of tumors without affecting normal brain functions. This is followed by radiation and/or chemotherapy. This is further specified by the STUP regimen, which is a protocol for radiotherapy plus concomitant and adjuvant temozolomide. However, available data on the FDA-approved drugs to treat glioblastoma has shown negligible improvement in survival, indicating an unmet need to explore agents with better therapeutic outcomes. The reasons responsible for failure of most glioblastoma treating drugs could be attributed to the physiology of the brain itself. Limited drug permeability to the normal brain is often due to the blood-brain barrier hindrance and the blood-brain tumor barrier in case of a tumor harboring brain. The complex tumor microenvironment also adds to the existing problem. Even diagnosis of glioblastoma using the current gold standard MRI could at times be inconclusive, as a mass detected on MRI could be an abscess or lesion, which could be misdiagnosed as a tumor. Biopsies are inherently invasive in nature, and analyzing dead tissue could lead to artifacts. Although PET scans offer guidance in surgery, radiation planning, tumor grading, and therapy monitoring, untargeted PET tracers use has been associated with off-targeting and unwanted background signals. Therefore, we and many others in the field believe that targeted delivery of radioisotopes is the way forward for superior theranostic applications. Engineered antibodies with superior antigen binding capabilities and low immunogenicity profiles have carved a niche for themselves at the forefront of modern medicine. In recent years, they have been popularly utilized as vectors and chemically modified further to carry radioisotopes for either medical imaging or radiotherapy, sometimes both. The targeting antibody, however, needs to be chosen based on the biomarker antigen it recognizes. These biomarkers might be overexpressed in the disease, but absent or poorly expressed in normal condition. Our collaborators at Gliotherics, an Australian immuno-oncology company based in Sydney, have identified a novel cell surface biomarker known as Glypican-1. Expression levels of Glypican-1 are elevated in multiple cancers, including glioblastoma. Gliotherics has also developed a clinical stage antibody miltuximab, which was well tolerated in a first-in-human trial. Together, we are developing and testing miltuximab-derived antibody radioconjugates, which could be useful in diagnosis and therapy of Glypican-1 overexpressing tumors like glioblastoma. Here I have highlighted preliminary results of the ongoing research towards developing zirconium-conjugated miltuximab-based Immuno-PET tracers. The long half-life of zirconium-89 matches the biological half-life of antibodies, and its decay characteristics allow high-resolution PET imaging. Post-desferoxamine or deethocylation and radiolabeling, miltuximab zirconium radioconjugate was tested in bulb-seen nude mice bearing subcutaneous glioblastoma tumors. Reconstructed PET-CT images at day 9 post-injection revealed maximum uptake in tumors in a Glypican-1 expression-dependent manner. tumor to liver ratio greatly improves at the endpoints of this longitudinal study. Autoradiography shows homogeneous distribution of the antibody radioconjugate across the tumor. Initial results from this preclinical study appear promising and can be explored in orthotopic models in future. We intend to further test antibody-derived shorter fragments and their feasibility for use as Immuno-PET agents. Outcomes of this study would be informative in delineating the impact of molecular size, avidity, the presence or absence of an FC region on the pharmacokinetic and biodistribution profiles of different formats. We have optimized enzymatic digestions to generate the Fab2 and the Fab fragments and recombinantly expressed the SCFV fragment. These are also tested for purity and immunoreactivity. The imaging data for these shorter versions are being processed at the moment and would be an interesting comparison with the whole antibody. I would like to end my talk by mentioning that although the brain is a very selective organ, there are new studies and clinical trials on ways to bypass barriers to protein drug delivery to the brain. Implantable devices like Ommaya reservoirs are more invasive than intra-arterial or focused ultrasound-induced transient blood-brain barrier opening. Nonetheless, these novel delivery schemes and strategies do encourage development of antibody-based radiopharmaceuticals against brain cancers in the near future. This concludes my presentation. Thank you for your attention. I would like to thank the organizers of ANZ-SNM to provide me the opportunity to speak to you today. And finally, I can't thank my supervisors and my collaborators enough for their constant support and encouragement. Thank you.